Welcome to the third webinar in our metagenomics webinar series. In today's webinar, we are talking about clean label formulations and using metagenomics as an approach to help scientists with reformulation and clean label approaches. My name is Joe Heinzelman. I'm the Director of Business Development for our Food Safety Genomics Services. Just a kind of recap of what the agenda is today. So we're going to do a little bit of a recap on some of the metagenomics and how that will help you with different product formulations. We're going to talk about experimental design and how the timelines and how you think about the experiment that you set up with a metagenomics shelf life study is important. And then we're going to go through an example of data and uncovering what it's telling us and what would be in one of these reports. If you haven't seen the other two webinars, we will recap briefly of some of the content that was in there. There's the first one that was done was a case study for troubleshooting spoilage, and we've got a bit of background in this one. I'd suggest you at least consider starting there. If you've heard a lot about whole genome sequencing and would like some clarity around metagenomics, shotgun metagenomics, and 16S metagenomics, I suggest that you start out with that webinar as well. And both of them are available on our website and on our YouTube channel. So just a couple slides of recap. One of the things I want to make sure everyone is really clear on, and this was covered in depth on the demystifying webinar, is that next generation sequencing is a big umbrella of technology that includes whole genome sequencing. The type of technology we're talking about today is specific to 16S amplicon sequencing, and we're using a metagenomic approach. And what that means is we'll be able to identify all the different types of bacteria in a sample. But this is just one application within the next generation sequencing suite of applications, including targeted amplicon sequencing, GMO testing, antimicrobial resistance, plant mapping with microbiomes, shotgun metagenomics. All of them fall within this bigger umbrella. And whole genome sequencing is an important aspect of next generation sequencing, including ID and relatedness. 16S metagenomics follows a fairly straightforward process, and this is a service that we are doing at our Lincoln, Nebraska genomics lab. We would receive samples in, and those include environmental swabs, or in the case of a shelf life study, finished product. We go through a extraction to make sure we get all the bacteria available and make sure that that DNA is available. We do a short PCR step. We're looking for the 16S gene, which is a conserved yet variable gene that helps us differentiate between different genus of bacteria. And at that point, we sequence everything within a sample. From there, we're using that data set and constructing reports that provide a lot of meaning and help lay out the data to generate some key action and key understandings. So just to kind of recap, what we're talking about is different than whole genome. With a whole genome, you're doing an enrichment, generating an isolate, and using that on a sequencer. And that's the next generation sequencing aspect of these services. You're building a genome of that one organism, and then you're using that for data analysis. With 16S metagenomics, we're taking a sample and we're extracting all the different bacteria from that specific sample, doing a PCR to amplify the gene of interest, and sequencing that for these different types of data sets. If you aren't familiar with shotgun metagenomics, it's kind of that next step, and it provides additional data. There are some trade-offs between the two methods, but it's important to know that with a full shotgun metagenomics, you can do both phylogenetic trees and relative abundances. So kind of put this in perspective, the two kind of techniques we're trying to differentiate here are whole genome sequencing. We're looking at building a data set of 3 to 4 million base pairs assembly on a specific organism. And with metagenomics, we're looking at hundreds of gene copies that are about 450 base pairs long. And so if you can imagine generating a copy of each bacteria's 16S gene and sequencing that, that's what we're doing. And it's a small subset of what's there, but it's enough information where we can do it quickly and accurately to identify the microorganisms. And then if you compound that with shotgun metagenomics, we're talking about much, much bigger data sets. So this is an example of a metagenomic output. And the key things that are important to observe is each bar represents a different sample. Each color represents a different microorganism. And the size of the color represents the relative abundance. So in this example, this was the example from the case study. And what we did here, where we're able to track this specific type of meothermus within our processing facility from different swabs, and we matched that to a finished product that helped us figure out what the problem was, how it was transferring within a facility, and that gave us enough information on how to fix it. So our subsequent data sets showed that we successfully eliminated, one, the source of meothermus and all the transfer points. So this brings up, it's important to kind of contrast how we're identifying these traditionally to put this into context. So for each organism that you would see on a specific bar, so for example, that S30 bar on the left-hand side, we can see 3 or 4 different types of microorganisms. If we wanted to identify them in a traditional sense, we'd have to go through, we'd have to grow them up. We'd have to do an isolation after we picked the correct colony. There's an initial slant and ID, which would also require some interpretation. And then there's a biochemical ID, which is used in a lot of different cases. And there are some other methods, and they all have trade-offs. But the point here is these are all relying on the growth and colony picking of a microorganism, whereas a metagenomic approach does not. And really what this lends us to is it's a new approach to microbiology. So a triptych soy agar, or a total plate count, is really a misnomer. It really isn't all the microorganisms. And what we're learning here is a lot of these different microorganisms that are growing on triptych soy agar aren't representative of issues or concerns or potential problems based on a formulation or a packaging environment. And so our challenge nowadays, as we move to the clean label, with these reformulations, what are we changing in terms of the growth environments? What are we doing to understand that? And as we gain an understanding of certain bacteria that are more prone for growth in a new formulation, what can we do to prevent that? So as we look at a traditional shelf life analysis, that includes aerobic plate counts in psychotropes, Enterobacteriaceae, lactic acid bacteria, Pseudomonas, anything else that would be relevant to that product. What we're using metagenomics for is get a more complete and clear picture of these microorganisms and how they change over time. And that's really the critical thing. And what we're seeing is that what's necessarily there from day one isn't necessarily the most abundant microorganism at the end of shelf life. Now we've got one test that does all the anaerobic microorganisms, all the ones that are viable and might grow in either a low pH formulation or a specific type of formulation but are harder to culture, various types of spoilage organisms, and also some of those low-level, slow-growing microorganisms that only show up after 30 days of shelf life. And so this includes some of the obscure ones that we've seen actually in some of these other metagenomic analysis that are hard to identify or are not part of everyone's database. So now we hopefully everyone's got a good understanding of the differences between a metagenomic approach and maybe some of the aspects to consider, why you'd consider metagenomics over a traditional shelf life, and how that's playing into the clean label formulations and the changing dynamics of both the food and the packages. So now as we get into experimental design, what we're really going to think about is comparing different formulations or different conditions in which the food is made or packaged. And there's kind of 2 things to remember. Certain organisms change over time, the populations and what they look like. And then one thing that we'll want to have within an experimental design is a control formulation. So whatever you're doing today are kind of your base. And there's 3 aspects that we kind of thought of as we were putting the slide deck together as to test. So maybe processing conditions might have a positive effect on shelf life. And so if you've got, let's say, a high pressure pasteurization application that you're considering, that would be a good control and variable to put through metagenomics. New additives that could extend shelf life or maybe a new additive that you think is limiting shelf life, a new flavor, a new ingredient, etc., or even new packages. And so, if you go to a modified atmosphere packaging, how is it changing the end shelf life microbiome? So this is a pictorial aspect of what we're talking about here. And so we've got a control group and then other variable formulations. And the really important thing that I want to bring up in terms of what we're offering is the ability to receive packages and shelf life study samples over time, and then generate one big report with this time-dependent microbiome component, which we'll show in the next few slides. And that's really important because, to be able to take a complete snapshot of a microbiome at day 1 and leverage that information against the day 30 would require an extraction and sequencing of everything. So we're set up to receive samples over time and sequence everything at the end to help reduce costs and generate more meaningful reports. So now we're getting into some data. This is from a peer-reviewed publication. I think it does a great job of really showing differences in formulation and how this is really important. If you can see that, there's kind of 3 different big colors that you see. One of them is Anoxibacillus, and that's the main gray that you see. There's a Geobacillus, and that blue one is a Serratia. And what we're going to see here is a different profile as we kind of generate these reports. So you can see as we progress from the initial time point, there are changes within each formulation. Formulation B has a lower abundance of the Anoxibacillus, and we're seeing Erwinia come through, which wasn't really apparent at day 0 in formulation C. And as we progress through the time, you can see this other microorganism coming up and dominating the profile in formulation A and B, but it's being held back in formulation C. And the green ones are Pseudomonas, and you can see on the left-hand side, the microorganism in the purple or maroon is dominating most of the profiles. Some of them have Erwinia, Geobacillus, and Pseudomonas. Now, if this was a moderate water activity product and we are getting Pseudomonases that can contribute to spoilage, and you could come to the conclusion that formulation C was contributing to holding back Pseudomonas more so than the other 2 formulations. Here it is again. As you can see, we're seeing similar profiles at the beginning. This change over time is very important. And then the other aspect is there are things showing up at the end of shelf life that wouldn't necessarily be picked up earlier in the product sampling process. And these changes are telling us things about the formulations that help with reformulation, understanding what to do or how to control it. And the other thing that would be very important is, let's say that formulation A is the new product and we have to address Pseudomonas moving forward. We could actually then go back into the various raw materials to see if it's a new organism coming in from a specific raw material or ingredient, etc., and provide that type of information to arm somebody with a new way of addressing that concern. The other really important thing that we'll see in a lot of these reports, if you were to go through with this, is what's called a principal component analysis. So this graph might seem daunting, and we're not all microecologists. And one of the things that we like to do to help people understand what's going on is provide some analytics behind it. And this one, after you look at a few of them, and we would help you get through the data sets, is that they're grouping the different samples together. And these groups of samples are telling us key differences between the different formulations or microorganisms or control versus variable. And this is helping us go in and look at specific samples to look at what is potentially the problem. So I know I said this earlier before, but these are being run at our NeoSeek facility. It's in Lincoln, Nebraska. It's actually the largest animal genomics facility in the world. This is where all the genotyping is done for a lot of the livestock. We do parentage and other various performance characteristics. If you're ever in Lincoln, Nebraska, feel free to stop by. We'd love to have you. And it's a wonderful facility to see a lot of this, the application of genomics for the improved efficiency of the food supply chain. So if this is something that looks kind of interesting, I'd highly suggest setting up a call with one of your reps and talking about what goes into the experimental design. We've got various sample submission forms that kind of help you keep track of that. And we incorporate the different groups within the data analysis, and that'll make interpretation a lot easier. We've got some recommendations in terms of how much product to include and how to go about that. Typically, after the last sample is received, we're on a 10 to 14 day turnaround time. And that's something that we'd be happy to talk about with you. So if you're not familiar with some of the other NeoSeek next generation sequencing services, the other ones include STEC ID and confirmation. So this is a 24-hour confirmation service we do for Shiga toxin producing E. coli. We also are doing salmonella serotyping. So instead of using traditional anti-sero, we're using a molecular serotyping method looking at specific genes that help us identify the serotype. And if you're not already using meat species identification for food fraud, that's a great way to help monitor and look at a supply chain that includes meat. If you have any questions, feel free to contact your rep or myself. And again, if you'd like to follow us or message us on any of the social sites, we'd love to have you there as well. Thank you, everyone. Happy New Year, and have a good day.